Biology, Chapter 19, Theory Video 12, Genetic Technology and Medicine. So obviously, because genes are quite directly related to many diseases, we can use genes to treat treat different conditions by changing the DNA, that as we discussed earlier, and that is actually the ultimate end of our studies. But before that, we can actually produce different products. We can develop or produce different products using the gene technology that we discussed earlier by using recombinant DNA techniques. So we discussed about producing insulin earlier. The insulin was produced using bacteria, yeast, and cultures of different mammalian cells. There are different advantages of using these things. The advantages are they have simple nutritional requirements, and they can be produced in large amounts. They can produce the product in large amounts, and also the production facilities do not require large space. You don't need massive factories to produce them, and also anywhere in the world you can produce them. So you don't have to have specialist instruments. You require specialist instruments, but they can be they are simple enough to be made anywhere. Then there wouldn't be any ethical or practical problems, because proteins they do not have to be collected from blood donors or animals by killing them or whatever it is. So we can simply produce them using these microbes. So the human proteins are produced besides insulin. There are different types of them. For example, factor 8, that is a blood clotting protein needed by people who are suffering from hemophilia. Then adenosine deaminase. This catalyzes the breakdown of deoxyadenosine in T lymphocytes. So the factor 8 is required for blood clotting. For people who are suffering from hemophilia, this cannot be produced. So as a result, this can be by using recombinant factor 8, they can be given this factor 8 regularly without having much problem. But before this recombinant DNA came into effect, it was given by blood donation. So that was a big problem. Now so this when there is a mutation in the F8 on the X chromosome that produces the factor 8, so that produces that results in the problem, hemophilia. So to avoid that, we can or to actually to live with it, to treat this disease, we can provide factor 8 produced by the genetically modified microbes. Then mutations of the gene ADA on chromosome 20 lead to deficiency of adenosine deaminase, ADA. So this causes a disease called severe combined immunodeficiency, SCID. So this enzyme will catalyze the deamination of adenosine and deoxyadenosine as part of the DNA metabolism in cells. Deoxyadenosine is toxic to T lymphocytes. So as a result, if a child is born without functioning ADA, because of the toxicity, they will die off. They have to be given regular injections of ADA. So this is also a possibility, because genetically modified E. coli bacteria and cell structures that have been taken from insect larvae would produce these ADA, and as a result, we are able to treat these conditions easily. Write down a note on this before we move on to the other part. Genetic technology and medicine. Genetic technology allows humans to use recombinant DNA techniques to produce different products such as insulin, factor 8, and adenosine deaminase, which are required for disease treatment. They have multiple advantages using these bacteria, yeast, and cultures of mammalian cells, such as the simple nutritional requirements, large amounts of products being produced, not requiring large spaces, and being able to be produced anywhere in the world, and having no ethical or practical issues of extracting blood from humans or extracting it from animal sources. Now we have to discuss about genetic screening. So now this is the analysis of a person's DNA to check if that person is a carrier, or if he has one or more alleles that would be associated with a disease. So they might not show any symptoms. So that is the reason why this is called genetic screening. Now, if a person is suffering from, if a family, if there are people who are in that family who are suffering from Huntington's disease, so they might not develop symptoms. So that person would be asked to, if a person is, having symptoms, that person would be asked to have a genetic test to confirm the diagnosis. So by genes, they can figure out that person has Huntington's disease. So then the other people also will be asked to have this test to figure out if that person is a carrier. So by using PCR to amplify the gene HTT and electrophoresis, they can determine the length of the two alleles and count the number of CAG repeats, and by this they can figure out if they are carriers. Then another disease is cystic fibrosis. It is another genetically transferred disease. The protein, the gene that codes for a transporter protein called cystic fibrosis transmembrane conductance regulator. So this is, the cystic fibrosis is caused by the recessive allele of that gene that codes for this particular CFTR protein. So this protein is a transmembrane channel protein, so that controls the movement of chloride ions down the electrochemical gradient. So when this malfunctions, there would be different cystic fibrosis. You don't actually have to explain what happens, the process, how it happens, but basically I will explain how it happens. So these chloride ions help the movement of water in tissues. So when there is, when it is required, where mucus is produced in these places, in places such as digestive and gas exchange systems, this water needs to be moved. So the cells in the airways, they have the highest concentration of CFTR because so this highest CFTR is actually present in the recently discovered pulmonary ionocytes. They have very few in numbers, but they have they are responsible for most of the movement of these chloride ions. So all in all, what happens is the if when there is a when there is a malfunctioning of this protein, so this movement is going to be a problem. Even with coughing, this cannot be moved. So the mucus will stay stick and sticky, and they cannot be moved. So the what happens is, to be healthy, a person might have this recessive allele and might not be able to might not show the symptoms. So if that person has a child with another heterozygous carrier, and if the child gets both the recessive alleles, then the child is going to have this disease. So to prevent this from happening, they do a screening test to figure out if the child has if they are carriers. So then there are BRCA1 and BRCA2. These are the genes, when mutated, will result in breast cancer. So they can also be screened by using this DNA technology. Then a parental diagnosis of a condition is actually, but let's write down a note on this first of all. Genetic screening. Genetic screening is the testing of an embryo, fetus, or an adult to find out whether a particular allele is present. For example, a person who is having Huntington's disease, and his family members would be asked to do a genetic screening to figure out if they are carriers of this disease. Cystic fibrosis and BRCA1 and BRCA2 genes in which mutations result in breast cancer also can be screened and figured out if they are carriers, and also if they have those mutations. Now, in parental diagnosis, what happens is, when a fetus has been developed, they can, within about 15 or 16 weeks into the pregnancy, or between 10 to 13 weeks of pregnancy, you can you can obtain amniocentesis. That is, amniocentesis, obtaining amniotic fluid, and then testing it to carry out testing the genes in them, or the genetic materials in them, to figure out if the fetus has any of the genetically inherited diseases. So if it is found, then those people, the parents have the choice of terminating the pregnancy or going for an abortion. So the second method, between 10 to 13 weeks of pregnancy, you can take chronic villus sampling. So this also, it is obviously taking fluid and testing it. So it is actually this is a little bit different from how it is done. In this one, sample of the part of the placenta called the chorion is removed by a needle. So this actually does not have a risk of uh, uh, making the harming the fetus, because when you carry out amniocentesis, there is a uh, risk of that the fetus dying. There is a 1% increase of the fetus having a risk of dying. Um, so so this is actually this is another use of this genetic screening, so the parental diagnosis. So this is so like I said, there would be and also the all of these activities are supported by ultrasound scanning. So they have the ability of seeing whether there is any damage that is being caused, and also so they can visualize the fetus, and if they when they locate the placenta, and they carefully take these samples out. uh, so these are quite important advancements that have been made, but one thing that needs to be remembered is miscarriage can occur where 2 to 3% of all the pregnancies can result in a miscarriage, that is the fetus dying. But amniocentesis can actually increase the risk of miscarriage, so there is a risk in that as well. Then also there is a non-invasive parental genetic screening, that is without using amniocentesis and CVS, they can take these fragments of DNA would be DNA of cells from placenta would get added to the would have the same genotype as the fetus, and they will somehow get added to the mother's circulation. If these can be taken out, then that can be tested for any genetic problems. Write down a note on this parental diagnosis. This is the practice of checking a fetus for any genetic abnormalities by using three common processes. Number one, amniocentesis. This takes a sample of amniotic fluid at 15 to 16 weeks of pregnancy. However, this has risk of increasing the risk of miscarriage. Number two, chronic villus sampling is taking a sample of part of the placenta called the chorion, 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 and is removed by a needle. Both these processes are monitored by ultrasound scanning to make sure no harm is done to the fetus. A non-invasive parental genetic screening that does not increase any risk of miscarriage is isolating fragments of DNA from placenta that gets added to the mother's blood circulation. This can be checked for any genetic abnormalities, and the parents are given the parents would be able to choose whether to have an abortion if the fetus is going to develop severe forms of diseases. Right. Please do question number 14 and 15. If you have any doubts, please ask me.